The central dogma explains how DNA turns into RNA in order to then be translated into proteins. RNA is transcripted from DNA by RNA polymerase, and proteins are translated from RNA by ribosomes. The proteins carry out the biological processing. Genes encode protein, and protein determines the cell function. The genes expressed in the cell determines what a cell does. Gene expression is used in all life forms. Each step in the central dogma has a point in which certain functions are regulated by affecting the production of proteins, and certain gene expressions are controlled and therefore do not appear. Not every gene product is needed at all times, as some could be harmful, and it could require a lot of energy to express every gene at all times. Regulation of gene expression is critical for the life forms to adapt to the environment.
DNA methylation is the attachment of methyl groups, which are a carbon atom bonded to 3 hydrogen atoms, to bases, such as cytosine, in the DNA. This process is facilitated by special enzymes that help the methyl groups attach to nitrogenous bases. This is an example of a methylated nitrogenous base. DNA methylation can serve to turn genes on or off. Often, genes that are not expressed in a particular cell tend to be heavily methylated in that cell. This could be an example of a gene in a cell that isn't actually expressed. This is because of the heavy methylation that occurs at all these points circled here. Sometimes, when methyl groups around genes are taken away, the genes can actually become active again. This is because the addition of methyl groups makes it difficult for enzymes involved in transcription to reach the DNA. Before RNA polymerase can synthesize an RNA, enzymes have to go in to kind of call the RNA polymerase there. So if there are less methyl groups, it's easier for these enzymes to get to the DNA. It is important to note that DNA methylation helps regulate gene expression before transcription. That is, before the DNA is turned into RNA.
DNA methylation is important to a number of cellular processes, such as during the embryonic stage of development. A lack of methyl groups in the embryo will lead to abnormal development in organisms like mice, embryos, or certain plants. It's also important for epigenetic gene regulation, which is regulating heritable alterations in gene expression that aren't actually harmful and are important to development. Also, once a cell is methylated, its daughter cells will usually get methylated as well. And these methylation patterns are responsible for the process of genomic imprinting, meaning that methylation can regulate if only one parent's allele is expressed for a particular gene. If your mother's allele is methylated, it won't be expressed in you at all, but your father's normal allele will still be expressed, so it's like you're haploid for that gene. For example, say your mother has brown eyes and your father's are blue. If your mother's brown eye allele has been silenced by methylation, you will only end up with your father's blue eye allele, and then you'll have blue eyes even though blue is recessive. But this process can be dangerous because if the one active allele that you get is defective, you won't have a backup from your other parent, so you could get a disease.